phagocytosis is a really important part of how this innate immune response is going to function. And when we've talked about it before, we've used two kind of steps: ingest and digest. So while those are still true, when we think about those two things, what is a better way of remembering that than eating, ingesting, and digesting cake? So I know that's not spelled correctly, but if you add that extra I in there, it gives you the 5 steps involved in phagocytosis. And so we're going to talk about each of these here. And so there's a verbal description here. If you notice, here's step 3, there's ingest. And the next one would be digest. Here it's switched to killing, but if you want to keep it digest, if that is helpful, you totally can. That would then change this to cade, which isn't as easy to remember as cake, but however you want to do it. And so this first slide is kind of a verbal description of what's going on. And then those same 5 steps are listed here. Again, there's your cake. And this picture is showing how the process is moving forward. And so what we have here is it's just a generic term we use. So a phagocyte is going to be an immune cell that can phagocytose things. So it can carry out phagocytosis. So that could be a neutrophil. It could be a macrophage. There's a variety of different immune cells that can carry it out. But regardless of which type we're talking about, any phagocytic cell is going to do this first step that's called chemotaxis. And so if you look at that word, it's got the prefix chemo relating to chemicals. And then it looks like taxis, which is what it is. And so hopefully you can think of movement with that. And so chemotaxis is defined as the movement of something either towards or away from a chemical. So this is going to be a chemical stimulus that then causes our immune cells to respond in a specific way. Now, so the general definition of chemotaxis is towards or away from, but we want our immune cells to obviously move towards a microorganism or an infection site, etc. So having them move away from it is not super helpful in fighting an infection. So the specific response here is what we call positively chemotaxis, meaning it moves towards the chemical as opposed to away from. And what those chemicals could be, it really varies. There's different types of chemicals that can be the stimulus that causes the movement towards a site. So it can be the microbe itself, so components of the microorganism. So parts of that microorganism. It could be damage to our own tissue. So imagine a cut in your skin. That damage is going to rupture the cells and their contents are going to be leaking out. And so those chemicals can serve as attractants to immune cells to help ensure that anything that enters that cut is going to be taken care of through phagocytosis. Complement is something that we've touched upon. We haven't gotten to a great definition yet, so I've just defined those as proteins that help immune response. And then chemokines are really specific. Those are chemicals that trigger kind of inflammatory responses. So those are chemicals that our immune system uses to help lead an inflammatory response, and part of that involves chemotaxis as well. So that's the first step. And so in this picture, you have this phagocytic cell. Again, it could be a neutrophil or a macrophage or any other cell that can carry out phagocytosis. And it's going to be moving in this direction towards where these microorganisms are. So that's step 1. Step 2 is adherence. And just as it sounds, the phagocytic cell has to physically come in contact with. So these pseudopodia can extend out and actually attach on. So you can see them physically starting to attach. That's adherence. And just remember that it is this step that some microbes have found ways of making more difficult. So bacteria have these things called anti-phagocytic factors like capsules, or they can have proteins on the surface like strep pyogenes or group A strep has something called M protein present. And both of those make it harder for immune cells to actually grab on or adhere. So the second step. So that's kind of the race the bacteria are looking at from their perspective. They're trying to make this more difficult, but the immune system has come up with ways of making this process easier. And so we call this process so if here's the microorganism, this label is an M, and here's the phagocytic cell. We have a way of kind of labeling this or tagging it to make it easier for our immune cells to adhere to that particular microorganism. And so those little stars that I'm drawing, their fancy name are called opsonins, so there they are. But you can think of them as molecular tags, or you can think of them as molecular highlights, or you can think of them as labels. Whatever works best for you to remember, but the term is called an opsonin. And what that allows immune cells to be able to better do is actually attach on to it during this adherence step. So bacteria are trying to make it harder. Our immune system is trying to find ways of making it easier. And so this is a battle that's going on. And as we learn, some bacteria are better able of avoiding this. And because of that, they're more virulent and are better able to cause disease because they can avoid our immune system. And more specifically, avoid adherence during the phagocytosis steps. Step 3 is that ingestion. So here's step 3, ingestion. And so what that means is the actual microbes are internalized. And so now they are actually inside the cell. And more specifically, they are enclosed within, there's different names for this. It's called a phagosome, or it's also called a food vesicle. So that's shown here, food vesicle, phagosome. And so that's what this structure is right there. Now going way back to when we were talking about cellular structures, there is an internal organelle in eukaryotic cells. So our cells have something called a lysosome, inside of which are digestive enzymes. And so normally they are contained within the lysosome, but the killing step or the digestion step, depending on which term you want to remember, is going to involve the fusion of those two things. Phagosome and lysosome fuse to become now a phagolysosome. And so those digestive enzymes are now going to be dumped onto the microorganism, thereby killing it. Right, so that's what we see here. Remember, some bacteria resist the digestion. And so bacteria have found ways to avoid phagocytosis. Some avoid being recognized and bacteria don't even move towards them, or some bacteria are going to be more resistant to the adherence step. Some are to this killing step. So it just depends upon the bacteria. But the goal is to obviously to digest and destroy the microorganism. And then step 5 is the elimination. And so the process is exocytosis. And so these would be the bacteria parts that are broken down. And so that's phagocytosis, but I just want to do an extension. Remember, macrophage is an APC, which stands for antigen presenting cell, and that was the molecular sketch of what that microbe was. So instead of just releasing these parts, antigen-presenting cells can not ignore that information, but use it and say, this molecule right here, that is an important part of this microorganism. So if you see it, you might want to attack it. So it gets a little more complicated, but that's the gist of at least the beginning stages of antigen presentation. The process of phagocytosis requires, as we mentioned, actually moving the microorganism inside of. And that's not the only way that immune cells can kill things. That's one really important way, phagocytosis. But what's listed here are ways that cells can kill things extracellularly, meaning keeping the microbe outside the cell, but then still having the capacity to kill it. So one example is eosinophils, and we said that those parasitic helminths, the worms that we saw, remember the adults can be quite large. So parasitic helminths can be quite large. If you remember seeing some of those adults, they can be, several feet long. So it's not possible for those worms to be internalized inside of a cell. So instead you have, so say this is the big worm, what you can have is a bunch of these eosinophils surround that worm, and then rather than trying to ingest it, they just release digestive enzymes directly onto the surface of that microorganism, of that worm, excuse me. So one thing that we can also see, those toxins and those digestive enzymes that are released can then weaken or kill that actual worm. And so eosinophils are not the most abundant type of cell. And so if we do have an infection that's from one of these helminths, one thing that we see is actually increase in number of eosinophils because they're needed to fight the specific infections. So eosinophilia or elevated eosinophil levels is one sign of actual helminth infection. And so this just shows this process. So this would be that helminth worm, and many of these are eosinophils, and you can see them starting to swarm. And then what you can't see is a release of chemicals, but you can see them move towards it and actually totally engulf it. And if all of those are releasing chemicals that are harmful, then as you might guess, that could be an important immune response that we see. Another example are these natural killer lymphocytes. So this is a category of white blood cell that we haven't talked a lot about, but lymphocytes, these NK cells, same sort of thing. They can, if we have a cell that has a virus in it, we can have these NK cells come up and potentially release chemicals directly onto that virally infected cell. Or another example is you have a tumor cell, you have these NK cells come and then dump contents onto them to kill those. So remember, in each of these cases, these are both human cells, but they're not normal. They're somehow abnormal. So there's viruses inside the cell. This is a tumor cell that's growing uncontrollably. And so there might be good reason for our own immune cells to kill our own cells, but there needs to be some way to differentiate normal cells from abnormal. And so NK cells have the capacity to do that and to target and kill those sorts of cells extracellularly. And then one more example, neutrophils. They do have the capacity to carry out phagocytosis, but they can also release chemicals onto nearby invaders. And so we've seen these before. Superoxide, that's peroxide. Hypochlorite is basically bleach. And then also neutrophils can release these longer fibrous fibers that can form kind of this, think of it as like a net that kind of captures. And then embedded in those fibers are some of these chemicals. And so it's basically like a poisonous net of sorts. And so it's important to realize that there's not one way that our immune system is going to be fighting something, but there is this a really robust and a really diverse way that our immune cell has mechanisms to protect us. And so it's not just phagocytosis, albeit that is a really important way. There are other mechanisms that cells have as well, and this slide indicates some of those. So phagocytosis is really, really important, but again, it's not the only way. And so we have cells that carry out phagocytosis, but those same cells can release chemicals that we saw that can be protective, like neutrophils releasing something similar to bleach or hydrogen peroxide, but they also can release what we saw earlier along those physical barriers, like lysozyme that can lyse bacteria, or those antimicrobial peptides called defensins. But there are some other chemicals that we haven't talked about yet, at least not in detail, like complement that I've touched upon, and interferon. So that's what we want to do over the next couple slides. So complement, what I've said, set of proteins that help immune system. That has been our working definition. And so that's still true, but we want to get a better definition and a more specific definition. So it's a set. So it's not one protein, but there's a variety of proteins. So serum proteins, so in blood. We can also call them plasma proteins because they're in that liquid portion of blood. And they are named, not very originally, C for complement with a number. And the number is just going to be based on the order in which they were discovered. So C1 was discovered first, C2, C3, C4, C5, etc. So there's upwards of 9 C1 through 9. We're not going to memorize what each of them do. There's several that I'm going to have you learn the specific function of them, but we're going to learn them as a group. That's what we're going to do. When complement is made, so when I'll say complement is made, it is inactive. So in our blood right now, we have inactive complement proteins that is found in that liquid portion of blood. And so this is describing two different mechanisms of how complement can be triggered to become activated. So that's what we're going to talk about first. And this is shown on the next slide. But before we do that, what I want to show you is how most complement become activated. So this is one complement. It's called C3, and so the third one discovered. And it's basically a big protein that has, I drew this line in it. And so upon activation, what happens is it's cleaved, that's a fancy word for broken, into two parts. So instead of having one piece, you now have these two separate pieces. And they now have the designation of C3A and C3B, indicating that original inactive form is now in the active form in those individual parts. So not all 9 of the complement do this in this exact way, but enough of them do that we can say it's kind of a pattern that we see. So many complement are going to be activated through this simple process of being broken into two parts, an A and a B form. And now each of those can go on and do something and have some function. So those functions we're going to discuss momentarily. But what we want to do first is talk about, well, what's going to trigger this activation event? And without going into too much details, what we're really looking at is what is the actual signal that's going to trigger it. So in one activation pathway that's called the classical pathway, it's the presence of antibodies. So again, remember, this is a product of our adaptive immune response. So this can take a little bit of time, but once our body starts making antibodies to an infection, one way that antibodies can be beneficial is that they can trigger the activation of complement that can then help the immune system. So that's known as a classical pathway. So that's shown here. Here is a microorganism. So this could be a bacteria, it could be a virus, it could be a fungus. There is some unique molecule on the surface called an antigen. And again, our body has responded by generating antibodies, again, from this adaptive immune response. And so when that happens, it's going to cause this activation event where eventually all complement become active. Many of them are activated through that mechanism we talked about being broken into two parts, and so there's a couple examples of it. Which ones do this and which ones don't isn't overly important, but the complements are going to become activated. And so that's known as the classical pathway. Okay. The alternate pathway is going to use a different signal. And so this doesn't rely on something that our bodies produce through the adaptive response, so not antibodies, but instead, notice these are going to be from the pathogen itself. So the pathogen or products from the pathogen are going to trigger activation. And so that's what the alternative pathway is going to use as its signal. And so realize these are going to be occurring at different stages of an infection. It takes 1 to 2 weeks for your body to make antibodies. So these are not going to be present early in an infection. And so this classical pathway won't be functional in activating complement early in infection. But as soon as the pathogen's present or products from the pathogen, this alternate pathway could be used to activate complement, so you can have active complement early in the infection through this alternate pathway. And that's what this is showing here. Here is again the microorganism. So endotoxin, different glycoproteins, and so those are again bacterial products. And that's going to again lead to all complement become activated. And so at the end, you have all the complement becoming activated. The only difference is what was the initial trigger. And so in one, it was antibodies. In another, it was bacterial products. Okay. So these are the only specific activated complement that I want you to know. And hopefully these functions mean something to you. So we've seen this term opsonization. So we can say C3b. So that was formed by breaking apart C3 into an A and a B form. And so this C3b is an opsonin. So that molecular highlight, tag, label, whatever you like. And that's going to increase step 2 of phagocytosis. So if you go back and look, that's cake. And so that second step is adherence. So being able to attach to the microorganism that's causing the infection and the disease. I'm going to jump down to chemotactic factors. So there are 2 here. The other half of C3, so this is the A form, and C5a are both going to be important for chemotactic factors. So again, step 1 is chemotaxis. And so this is going to enhance step 1 of phagocytosis. These same chemotactic factors, so notice it hasn't changed, it's C3a and C5a are going to enhance inflammation. So we haven't talked about inflammation yet, but we will. So look at those later slides and we'll talk about inflammation. So those are some individual functions of activated complement. So C3b is an opsonin, and then realize, well, why is that beneficial? That is going to lead to enhanced phagocytosis through increased adherence. Chemotactic factors, C3a, C5a, that's going to be important because it's going to enhance phagocytosis through increased chemotaxis to that area. So more recruitment of immune cells to that area. So that's important. And then while we don't have a complete picture of inflammation yet, just you kind of have to take my word for it that stimulating inflammation can be beneficial, and we'll see that. So those are some of the functions. One last function. Oh, it's here. And I guess this is on the next slide. And so one last function in addition to this is what's called a membrane attack complex, or MAC. And so in addition to helping out phagocytosis and being kind of, like I defined before, helper proteins, these have a direct effect on bacteria that we're going to talk about. So that's shown here, so I just wanted to bring it up to you. So this is describing what are the functions of activated complement. And so we've seen C3b is an opsonin, C3a, C5a, chemotactic factors, inflammation mediators, C3a, C5a. What we're going to break down inflammation in a little bit. There's two really important changes that occur in the tissue, and so those are seen here. So again, you can just kind of put a note, see inflammation. So by the end of this lecture, we will have a better understanding of inflammation, but there's two major changes that we see happen. Increased vascular permeability. That's a fancy term to describe leaky blood vessels. And then there's that diapedesis term again. That is the ability of white blood cells to leave the blood at specific regions. Okay, so I'm not going to talk a lot about that now, but we'll see it momentarily. And so what we really want to focus on is this role, because we haven't talked about it, and this one that I just mentioned. So one other really important function of activated complement is as an enzyme to activate other complement. And so let me show you what I mean by that. So this picture it looks really complicated, but we're going to break it down into individual parts. So notice here is the pathogen. So say this is a bacteria. And this is going to be later in the infection. And the reason I know that, later in infection, because our body is already making antibodies. And so we have the antibodies binding. And so this, in this example, this is classical activation of complement. So if you go back and look, what is the signal or trigger of classical activation of complement? It is the presence of antibodies and them binding to antigens on the microorganism. Okay. So in this case, you don't have to know which number it is, but it shows one of the complements. It happens to be C1, goes from the inactive form to the active form. And then that active form is now an enzyme that can actually do that reaction that we said before. So here is another complement that is in the inactive form, but this active complement can break it into two parts, an A and a B form. Oops. An A and a B form. And it can do that for several. So here's an A and a B form. And now you have these parts that weren't present before that can interact in specific ways. So here you go. And now you have a new enzyme that comes together and that can split these guys into an A and B form, and now you have all these other fragments that can come together to form another enzyme. And so this is why we call it a cascade. So you have one triggering event, but the rest is kind of automatic, meaning that this is the only kind of thing that you have to do. And so you can think of it as dominoes. So as long as you have everything set up correctly, all you have to do is push one domino and start it to fall, and all the others are going to fall subsequent to it because of the arrangement of how this works. And so this cascade, we use that term to describe things like clotting cascade, where you just need one initial event and then all those subsequent steps will follow after it. And so that enzyme is really another important role as we see. The last function that I want to talk about is these membrane attack complexes. And so that will be visualized here as well. We have opsonins. So here's C3b as an opsonin. Here's C3a. That's enhances chemotaxis and inflammation. Here's C5a. That also enhances chemotaxis and inflammation. So those are seen as well. And so really the last thing that we haven't talked about are membrane attack complexes. And so this is a combo of activated complement. It tells you what it is here, but you don't have to remember what it is, but you can see the different complements. So it's C5b, 6, 7, 8, 9. But what's most important to realize is what the membrane attacks complex forms is a hole in bacterial membranes. So there's a hole, and that leads to lysis of the cell. So these are supposed to represent a bunch of holes formed by the membrane attack complex that comes from activated complement. As this illustrates here, gram-negative bacteria are going to be more susceptible to these membrane attack complexes than a gram-positive bacteria. And so I pose this, I'm going to do a brief pause and see if you remember, but the short answer is, or the hint is, it relates to the cell wall or the cell wall differences that exist between. And so to answer this question, you have to remember, what is the difference between a gram-negative and a gram-positive? And so the first thing I want you to remember is peptidoglycan differences. So one of them has a thick peptidoglycan and one has a thin. You now have to remember which is which. And so the gram-positive has the thick peptidoglycan. So I don't know how many times I've drawn this, but this is supposed to represent the thick peptidoglycan layer. Okay, and this one has just a thin. And then the other question is, which one has an outer membrane? And we've seen this at different times. Only one of them has an outer membrane. And that one, I'm just going to draw it as a dotted line here. The gram-negative has an outer membrane, whereas the gram-positive does not have an outer membrane. So now going back to this membrane attack complex, you have to think of these are a series of activated complement that need to get to membranes to form holes in it. So the only membrane that exists here sits underneath this thick peptidoglycan. So those complement have to work their way through the peptidoglycan and then assemble to form holes. So it can be done, but it's not as easy as, look, here is a membrane it can start forming holes in. That's the outer membrane. So that starts to disrupt the bacterium. Then once those holes are present, the complement can go through those, can get to this other cell membrane that sits underneath the the thin peptidoglycan layer. The next chemical we want to talk about is another protein. So just like complement, this is a series of proteins. These are collectively called interferons. So we're still talking about the innate immune response, but interferons are going to be part of the innate immune response and non-specifically respond, but to just a certain category of viral infections. So many of the other processes are more generic and generalized against different microorganism groups. But interferons are going to be specifically helping protect us from viral infections. One other thing to realize is that it is the production of interferons that can actually lead to us feeling a little bit, feeling the way that we do during a particular infection. So it's not necessarily the virus, but it's our body's response that makes us not feel great. And so interferons can be responsible for that. There are different categories of them. They're named for the Greek letters alpha, beta, and gamma, but we're just going to learn them collectively as interferons. Okay. So it does get more complicated than the way we're doing it, but so we're just going to look at interferons in general. And what they're going to do is you can think of interferons as basically a warning signal of sorts. And so we're going to look at this. But when a cell is infected by a virus, that virally infected cell is going to release interferon, and that's going to then serve as a warning signal to change the physiology of those neighboring cells. And this is the way in which they're going to do it, but rather than talk about it here, we're going to look at a picture to better understand how interferons can be protective. So here's the picture on the right. And we have a virus that has invaded our body, and not only that, it has successfully infected this cell. As we have learned, the goal for the virus is to make more viruses. Well, make and release more viruses. So that's what the virus's goal is. Before that gets fully in effect, what this cell can do is actually start making interferon and releasing it. And so again, that is that chemical warning signal that I talked about before. And so neighboring cells realize this is, no virus has infected this, at least yet. And so it has a little bit of a head start because it now has a warning that there is a virus in the neighborhood, so to speak. There's a virus in a nearby cell, and it is possible and likely probable that a virus may enter this cell in the near future. And so the way in which this neighboring cell responds is by manufacturing what are called antiviral proteins, or AVPs for short. Now, when they are made, when these antiviral proteins are made, they are inactive. So they are not active yet. And what's important to note, will only activate when virus infects the cell. And so that means there needs to be some sort of uniquely viral signal that will indicate that there is indeed a virus present within this cell. And what actually is often used to activate these antiviral proteins is something that is uniquely viral, which is double-stranded RNA. So our cells, human cells and most cells, do have RNA, but they're only going to have single-stranded RNA. So double-stranded RNA is unique to viruses. And so because of that, that is a really good signal that has kind of evolved for our interferons or these antiviral proteins to respond to. So if a virus does enter, and more specifically, there's double-stranded RNA, that's going to cause the AVPs that were inactive to now become active AVPs. And now we can look at what happens, or what is the function of active antiviral proteins. But basically, what's going to happen sorry about that, jumping around too much. Active antiviral proteins are going to stop protein synthesis, or another way of saying it is stop translation. And the way it does so is it's going to degrade messenger RNA. These antiviral proteins that are now active are going to bind to ribosomes and stop that protein synthesis. And so what that's going to do is stop the translation of viral proteins. So another way, stop virus synthesis. But at the same time, translation is also needed to make normal cellular proteins. And so this is not without any harm to the cell, which is the reason why when these antiviral proteins were made, they were inactive, because we didn't want to shut down the cell immediately. We only wanted to get ready. If that virus arrives, then at that point we shut down the cell to prevent the virus from making more virus. But this cell can go around and do its normal operational activities, and if the virus never arrives, then that's great. There was kind of no harm. And then those antiviral proteins will degrade over time. So it works out. But it's an important early warning signal, this interferon that's released, to get the cell ready for an incoming virus, if that virus ever arrives.